So before we get into our topic for today, there's just a couple of announcements from us on behalf of the Biocurator Workgroup series. So firstly, to say that this will be our last meeting before this meeting series breaks for the summer. So we're not going to have meetings in July or August. So please enjoy those couple of hours back to your schedule over those 2 months. Use them to do something nice to enjoy the summer. Don't know if I can give you that guidance. I'm not your boss, but we hope that you'll enjoy that time back to enjoy the summer. But over this time, we will be sending out a survey just to gather your feedback on the Biocurator Workgroup meeting series over the last sort of 12 to 18 months and see if you have any suggestions for how we could improve this series in future, any topic suggestions you may have. So keep an eye out for that survey coming at some point over the summer. And we'll be reviewing those results from September onwards. But as I said at the start, that this will be our last meeting and we will reconvene on September 10th. But with that, and we've got a good number of participants online, so I think we can dive into our topic for today. So today we're joined by Marina Distefano today with her GenCC or the Gene Curation Coalition database hat on, not her chair of this meeting series hat. And so, yeah, Marina's going to be giving us an update on the GenCC database. I'll hand over to you.
Great. Thanks, Alicia. I will share my screen. Of course, I had to get a package, right? Because we're going to share a screen here. Just going to take 2 seconds to throw my dog out. Okay, great. So today I'm going to talk to you about GenCC, and we give updates on GenCC, once a year, once every year and a half. So if you've been a biocurator for a while, some of this might be a repeat. However, I am excited to present some of our draft mechanism framework work today. So that'll kind of be new and hot off the presses. And this is our genetics and medicine marker paper here, in case you're interested in reading that. That was published a couple years ago. So I don't think I have to tell all of you, because you're doing curation, that standards for curation are very critical. But often when I present this database to others outside of ClinGen, I really need to drive that point home. This graph is a little bit old at this point. But when we went to the Genetic Testing Registry, or the GTR, which is the public registry that is required of all clinical US-based labs to register their tests, and we searched for the phenotype or the condition hypertrophic cardiomyopathy and plotted the number of genes from 14 different US clinical labs. I was wildly surprised, or maybe I shouldn't be, that they varied extensively, anywhere from under 20 genes to over 100 genes, just with the same search terms. So really pointing out that standards and consensus are needed for which genes are valid disease genes that are ready for clinical testing. And that's really why you're doing what you're doing, especially if you're doing gene curation, to try to provide some standardized curations for people to use. And add to that an additional layer that different levels of evidence are needed for different clinical uses. So you need a really high level of evidence for predictive tests, because people might be going and doing prophylactic measures such as surgery, based on your results, compared to diagnostic panels or exome and genome analysis on an affected individual where the prior probability of a genetic condition is much higher. And so this is why we formed the Gene Curation Coalition in about 2017 or so now. And that's the GenCC. And really our goals were to bring together a bunch of curation groups that either curate publicly, like ClinGen, OMIM, and the panel apps, and some of the private clinical laboratories like GeneDx, Ambry, LabCorp, formerly Invitae, Quest, and so on, to start to harmonize our curation approaches and also to share our data. So really the goals from all of these members of the GenCC, which you can see all their logos here, was to understand the approaches and classification systems of the different curation efforts, because while we were working in the same realm, we were maybe looking at things slightly differently. And for comparison, we have to figure out what our differences were. And then really to try to develop consistent terminology, starting out with the clinical validity strength, and then moving on to inheritance, allelic requirement, and then finally I'll tell you today about mechanism of disease. And then really with the goal, once these things are understood and harmonized, to post gene curations from any group willing to share with the public. So our database, and I'm sure many of you are familiar with it, is like a ClinVar of assertions, but for gene disease relationships. And so once we had that database stood up, which was maybe 4 or 5 years ago now, the goal was to resolve differences and collaborate on gene curation projects to try to bring a harmonized front to the field so that they wouldn't be looking at discrepant curations. So when we first started, we wanted to do a bake-off where we had many of our original members of the GenCC look at the same genes and curate them in whatever definition they decided was curation. And we found that different groups were doing very different things. For example, OMIM and Orphanet really were curating based on the presence or absence in their database. So does something have enough evidence to warrant even being present in their catalog of diseases? Or should it just be a gene entry that doesn't have a disease associated with it? ClinGen, is really evaluating a gene, a disease, and a mode of inheritance and the strength of that relationship. Panel App is asking the question both in Australia and the UK, like, should something go on a panel related to a particular indication? So it's just really different questions across some of these efforts. And you can see green kind of meant that there was a high gene disease relationship or something should be on a website, and red and like yellow oranges meant that there was like a moderate to low relationship. And you can see things were all over the place when we did this curation bake off in the beginning. So we really realized we needed to harmonize our approaches and our terminology. And the reasons that and I spoke a little bit to why there are so many differences, but the reasons that there were differences were the definition of disease. So sometimes groups were really looking at high penetrance conditions and monogenic versus inclusion of lower penetrance phenotypes. The purpose of curation, like I just talked about, should something be on a panel versus what's the validity? And then also differences in disease or phenotype assignments. So maybe they were performing their lumping and splitting differently, or they were looking at one limited gene disease relationship for a gene, and ClinGen was looking at a definitive gene disease relationship for the same gene, because there are multiple conditions that can be associated with a gene. And also, very critically, the date of evidence evaluation. We know that things are published all the time. So if someone's looking at an older body of evidence compared to a newer body of evidence, there might be discrepancies there. So we realized once we understood these differences and trying to focus on resolving them, we needed to harmonize our terms and our definitions so that we were all speaking the same language. Because how could we stand up a database if we were all meaning different things when we were classifying our gene disease relationships? So we went to compare our terms, and we realized, much like the bake off, that our terms were all over the place. So you'll see that a couple different efforts use colors, and they actually use colors to mean opposite strengths. For the gene disease map, which was just an effort based out of the UK, they used red for meaning really strong gene disease relationships, whereas panel apps use the traffic light system. So red means very weak relationships. So that was confusing. OMIM and Orphanet were using presence versus absence. ClinGen was using their terms. And G2P was using kind of confidence terms, essentially like probable, possible. So we were all over the place, and we really needed to harmonize that. And so what we did, and this is published about in the marker paper, is we came up with a Delphi survey process. And a Delphi survey is great in that you have iterations where you kind of throw as many terms out there as possible and slowly narrow down to the least unpleasant option, which sounds terrible, but that's kind of the best way to get to compromise. So in the first phase, we developed harmonized definitions because we needed to make sure we were all talking about the same thing. And these definitions did not contain the proposed clinical validity terms that were drafted for all the categories, because you can't circularly use those terms to describe the gene disease relationship bucket. For the second phase, we prioritized. So we solicited term suggestions from all GenCC members. And then we surveyed them. So 33 individuals filled out the survey, and they used the Likert scale to express their level of agreement, disagreement, or neutrality for these terms. And they were prompted to add additional terms in this phase. And then in the third phase, consistent with the Delphi survey process, we refined. So we used iterative surveys to refine the term list. So the second survey included some curation staff from the GenCC members. And that was 38 individuals. And after this survey, we started to whittle down the terms. So we um, attached numbers to the Likert scale. So positive numbers were for agreeable terms, and negative numbers were for disagreeable terms. And so we removed terms with scores that were less than, or that were greater than two standard deviations below the mean score, meaning that they were highly disagreeable. Um, and then for the third survey, we reached out to the international genetics community and we got 241 responses, and we surveyed to determine the final term list. And we worked together to record a 10-minute explanatory video of what the GenCC was and why we were doing this. And for phase four, we looked at consensus and then we implemented. So surveys 2 and 3, so the GenCC and extended curation staff and the international genetics community had 93% term agreement, so 13 out of 14 terms, suggesting that the choices that we made are in agreement with the larger genetics community. So all of the terms now are used to standardize submissions to the GenCC. We did add one additional term, supportive, for less granular submissions from OMIM and Orphanet, because as I mentioned, it's really just presence or absence in their database that is their curation style. And so they really couldn't go as granular as like strong, definitive, limited, moderate. And so these are the terms right here. And you'll notice that ClinGen had already largely used those terms. And then we mapped to no known disease relationship, which was a slight switch in wording. And then there's also a subtle additional category of animal model only when there's no genetic evidence at all, but there is an animal model because the genetics community found that was useful. So here are the statistics currently for the GenCC database, and the URL is search.thegencc.org. I'm sure many of you have used this in your gene curation efforts. And at this point, we have over 21,000 curations and over 5,000 genes from 14 submitters. And here's the spread of curations, with many curations being strong and definitive. There are some clinical lab submitters who will only go to strong, so that's why it looks like there are so many more strong curations when you would see in ClinGen, like we actually don't have a ton of strong curations because most of ours meet definitive if they're strong enough to meet strong. Also, Orphanet has a very large bolus of submissions. So there's a big chunk of supportive submissions in here. OMIM is still not currently in the database, but we're hoping to import them soon. They really wanted a more automated way to submit to the database because everyone does it in a spreadsheet fashion right now, kind of like ClinVar. And they wanted a more automated way to submit to the database because their database changes all the time. And that would have been really bifurcating it to have to like submit spreadsheets all the time. So that's why they're not currently in the GenCC database. And just a couple screenshots if you're not familiar with the database. This is the gene list landing page if you go to search.thegencc.org, because this one is like the statistics page. You'll see all the list of genes, and all the boxes along the top are searchable. So you can search by gene symbol. Most of the time when I'm doing exome or genome analysis, that's where I go and I just search for the gene. If you want to try and make panels for a particular, or like gene lists for a particular condition, you could search by disease. I will say this is an exact search, so it's a bit imperfect at this point. We're hoping to make disease searching a bit more flexible, but much like on the ClinGen website, this is a little difficult because people are using synonyms for diseases all the time, particularly within GenCC, where we have 14 different submitters, and many of them have their own ontologies, like OMIM and Orphanet. So you can search by that. You could filter by a particular submitter, or if you want to see a certain strength of gene disease relationships, you can click and unclick any of these boxes here. And if you have any questions about these strength buckets, you can click on this info button and it will actually give you the harmonized definitions that we came up with in our Delphi survey. So the original thing that you'll see is the collapsed line here, and you'll see counts across all of these different buckets, and we have very consistent coloring. That's the website team for ClinGen and the website team for GenCC are the same, so you'll see some overlap visually for this, but we have very consistent coloring throughout the website. And so you'll see in the little number circles here, like for example, this one, before you even would open it up, you would know there's one no known disease relationship curation here. And you'll see the number of disease equivalents. I'll talk about that in a second, and the number of submitters. And then if you want to know more, you would just click the open, like hit one of these little gray arrows, and it'll give you just kind of a quick sense of what's going on. So who submitted, what is the classification, and what condition is it for? So when we say disease equivalence, we map all diseases to the monarch disease ontology. We have to be able to accept multiple different ontologies because we have OMIM and Orphanet as members of the GenCC, so they have to be able to submit their own ontology. But Mondo, much like in ClinGen, organizes all of those disease terms in one ontology, and so we map everything to the monarch disease ontology to calculate disease equivalence. So in this case, there's one disease equivalent because everyone's submitting on Noonan syndrome. And ClinGen could have submitted on the Mondo ID for Noonan syndrome, and Orphanet likely probably submitted on the Orpha ID for Noonan syndrome, and they just get mapped together under the Mondo term for Noonan syndrome to give you that sense. And if you want to learn more on top of this, because you don't see the mode of inheritance, you don't see any of the evidence, you would click the details button here. And this will take you to the gene level page. So you'll see the gene on the top and the HGNC ID. And then there's a couple different ways to visualize the data. So the first one is by classification, where the strongest classifications will be at the top. And you can filter in and out different things on each page, like if you only want to see certain submitters. If a page is really, really busy, oftentimes I'll filter on mode of inheritance. If I only wanted to see autosomal dominant, for example, versus the autosomal recessive, you could click into that there. You could also do diseases or classifications if you only wanted to see definitives, for example. Um, the next tab is by disease. And this gives you a little bit more information. And this is a bit of a messier tab because, as I said, everyone submits using their own gene disease relationships, and they might be synonyms. They might be higher in the Mondo tree for one submitter versus lower in the Mondo tree for another. So you'll see what they originally submitted as. For example, for Orphanet, they submitted as the Orpha ID here, and then it's mapped to this Mondo ID of arterial calcification of infancy, um, which is also the same for TGMI G2P. However, you can see they submitted using the OMIM ID. So we're really trying to map this as much as possible to simplify a complex situation using the monarch disease ontology. And of course, ClinGen just submits Mondo directly. And then if you click into a particular submission, so you could click the more details button here, you'll go to a submission level page. And different submitters will have different pieces of evidence. So much of the Illumina curations actually have evidence summaries, a la ClinGen evidence summaries. Pretty much all of the submitters have PubMed IDs. So I do think it's useful to click into the submissions. If they have a public website like the panel app websites or G2P websites, they actually will include links to their publicly available data. And so that can be really helpful too. So, for example, Panel App will link out to their pages where you can go and see their additional information. So, oftentimes people will be in favor of linking directly to their sites to keep up to date instead of including things in a kind of static fashion in their submission. So, for example, ClinGen just links to their gene disease relationship pages so that you can see like the very granular table that we expect and the evidence summaries in that way. I think in future iterations, we might at least pull the PubMed IDs and the evidence summary into ClinGen submissions in the GenCC database. But right now, we just do link outs. You also can click and see the assertion criteria, much like ClinVar, it's required to have assertion criteria in the GenCC. So if you're not sure what a group is using to classify their gene disease relationships, you might want to take a look and see how they're classifying them. Because while everyone is required to map to our terms, they still are, as long as they can granularly map to these terms, able to curate in a way that they find is most useful for their resource, and just has to be approximately as robust as the ClinGen system. And then if you go to a particular submitter, um, so I think you can click the submitter up here, and it would take you to a particular submitter page. So if you ever had a question about a GenCC entry, we actually have a public contact for every single submitter, including their email, and they know that the email is publicly available. So feel free to email them if you have a question on a particular submission. We have a little summary of what their resource is about, a visualization of all their classifications, the link to the assertion criteria, like I said, and then a link to their website. So that's what's going on with the database, and all the data are downloadable. I didn't say that. We're hoping to have an API sometime in the future, but there are a couple different ways that you can download the data. And the GenCC data are also displayed in a couple different places. They're displayed on the ClinGen gene facts page. They're like open drop down if you do the gene search, and you can see like a little colored summary of the number of um, submissions for a particular gene in the GenCC and click to it. It's also displayed as a track on UCSC, and it's finally also displayed as a track in Decipher. And many people for their pipelines ask how often we update the data, and I would say most submitters submit quarterly at this point in time. We're hoping to launch a submission portal very soon, and so the data might be updated more regularly then. So you would maybe potentially want to check back if you're using data downloads at this point. You will also notice that ClinGen, as of right now, still does large submissions to the GenCC database. So you may notice a discordance between curations on the ClinGen website and curations on the GenCC website, because it's not automatically when you publish a curation on the ClinGen website that it gets sent to the GenCC website and displayed there. We are hoping very soon that that will happen like nightly, essentially, and then they'll be much more harmonized. But oftentimes when I'm doing exome or genome analysis, I will often search in both places just to be sure. Because as you know, ClinGen's publishing things every day. So now that we talked about the database, let's talk about what we're using the database for. Um, and much like we try to resolve conflicts within ClinVar with the VSEPs and with ClinGen curation of ClinVar, we're trying to do the same thing with GenCC, although we're largely doing it internally because it's a controlled database. So you have to apply to be part of the GenCC. And so for this reason, we're doing our own conflict resolution. And here are how we define curation conflicts. And this is consistent with the Bean et al. Paper that suggests that gene disease relationships that are moderate and above should go on clinical testing panels. So anything, any curation record that's moderate, strong, or definitive, we say is concordant. But if you have an entry where someone says moderate and someone says limited, we say that that's discordant because moderate says it should be on a panel. And of course, you have like a matched gene disease relationship. It's not like you're talking about a different um, disease or something like that. It has to be matched. Um, Moderate ones would say that if someone submitted moderate, it's saying that they should put something on a panel. And if someone submitted limited, they are saying that it shouldn't be on a panel. So that's kind of the line of conflict there, that all of these assertions are concordant and all of these assertions are also kind of concordant. And the ones that we're leaving out are those less granular submissions from OMIM and ORPHANET of supportive, because you can't really compare those apples to apples. So here is our resolution workflow, much like ClinVar resolutions, where we try to put it upon the submitters to resolve simple discrepancies, and we use some of the calls to discuss complex discrepancies. So our website team generates automated conflict reports. And as of right now, the simplest way to do this is to match the gene, the disease as an exact match in the mode of inheritance, and say someone said definitive and someone said limited. Um, we do try to work through some of like the synonymous disease terms, but as you can imagine, that's much more manual because it takes someone like me saying hearing loss is the same as hearing loss number X. And they're just different pieces in the tree. So we start with exact disease matches for these automated conflict reports. And then I usually review them manually and determine which ones are simple conflicts and which ones are complex conflicts. And what I mean by simple conflicts is if you have multiple submitters, potentially one of them is very out of date. Um, and you would go to that person and say, can you please re-review this? Your curation is 3, 4, 5 years old, and the other curation is brand new. And we suspect this is why there's a conflict. Um, Another simple one is an outlier submission. So if we have three submitters, for example, and two of them are saying um, strong and one of them is saying limited, we go back to the outlier submitter and say, can you please take a look at this? The other two submitters have said strong, and reevaluate. And if that doesn't solve it, then we would bring it like a more complex one. So this causes the generation of a submitter-specific conflict report for offline review, and then the submitter will update their own GenCC database submissions. For the more complex conflicts, so if all of them are submitted around the same time, or we really are split across the board, we're like two people are saying that something is definitive and two people are saying that something is limited, which is like a huge discrepancy. We want to discuss those during a GenCC meeting to make sure that we're all looking at the same data, and it's not like the definitive ones had some very large stash of internal data or something. And make sure we're on the same page. And so what we do is we consensus vote. So I present those complex conflicts and we'll try to pool the internal data from the submitters, particularly if they're clinical labs, although most of the time people are largely using published literature. And then I'll present that during a GenCC meeting and we'll consensus vote. And because we didn't want people to have to be told, like, you have to classify this gene disease relationship as moderate, you have to classify this gene disease relationship as limited, we vote in kind of like above and below ways, if that makes sense. So something can either be in a vote limited or below, meaning it shouldn't go on a panel, or moderate and above, meaning it should go on a panel. And that, in theory, should resolve the conflict because that's really what we're deciding is the conflict. Should something go on a panel or not? So here is a summary of some of our complex conflict voting decisions from the last couple years. And you can see that some of these really are complicated because some of them are resolved as moderate or above. We have 16. Some of them are resolved as limited or below, but others were so complicated and borderline that we actually decided to leave the conflict to show that there really is borderline evidence in the field right now. So six of them we actually left as the submitters may maintain their conflicts because this really is borderline evidence and we'll reevaluate later. And another harmonization effort I wanted to talk about before jumping into mechanism curation is after this, we then harmonize terms for variant effect. And so um, Ang Roberts had brought together the sequence ontology team along with the GenCC team to really come up with some terms for variant effects. And you can see some examples here. So the paper in Genetics and Medicine really talks about this in detail. And I think the cardiovascular ClinGen domain helped with a lot of this curation of testing out this type of framework. But so you can see some of these terms that we have. So for example, splice region variant, splice acceptor variant, if it triggers NMD, if it escapes NMD. And of course, there are different levels of specificity depending on the type of information that you have. And she made this really beautiful chart that talks about the predicted functional consequence that's in the paper, with blue meaning almost never, heading towards yellow meaning almost always. So, for example, if you have a splice acceptor variant that triggers nonsense mediated decay, it almost always leads to a decreased gene product or absent gene product, and rarely has like no effect. So this was a really helpful terminology paper, and the variant effect predictor also team worked with us too to potentially adopt some of these terms. This was largely expected to be used not by manual curators, but by those programs that annotate the effects of variants. Okay, so we have about half of our time left. So I want to tell you about something that is still in draft. So it may change. We're working through piloting it now. But this is our work that we've gone through developing a mechanism curation framework. So over the past 2 years, we've iterated upon a semi-quantitative mechanism framework. This framework includes the mechanisms for loss of function, gain of function, dominant, negative, and specifically not loss of function. Maybe you don't know the rest of the mechanism, but you know that loss of function is not a mechanism of disease. So importantly, the loss of function frameworks will be adopted by the sequence variant classification version 4 group for their guidelines and used in their variant effect flowchart. And so we're really trying to very thoroughly vet these frameworks. And also excitingly, the ClinGen dosage sensitivity working group has already begun in February this year using the biallelic loss of function framework to curate semi-quantitatively strong definitive gene disease relationships. Because you'll notice in the past, dosage curation just had 30 autosomal recessive as a phenotype. So there wasn't really additional information about the strength of a particular mechanism for autosomal recessive. It was just it's autosomal recessive. So I'm going to take you through some of the draft frameworks and give you examples of curating through those frameworks. So the first one for loss of function, we've split it into biallelic and monoallelic loss of function because some of those have different considerations, as you would note from the dosage curation framework. And I would say much of what we have used in our scoring has been taken from the dosage curation framework because they were really thinking about this first. So, for each framework, to get you in the right mindset, we have gene level questions. And many of them are similar, really just to get you to think about and many of these questions are questions that you would ask yourself when you're doing the PBS1 flowchart or when you're going through the variant effect kind of flowcharts in the new guidelines to flag certain regions that might be concerning where loss of function might not actually be the mechanism of disease. So the first one is what's the size of the gene and the number of exons, really to give you a sense, like could nonsense mediated decay be predicted or is there just one exon? Is alternative splicing predicted? Are there certain exons that you should be warned about that are spliced out in biologically relevant transcripts, such that if you see a loss of function variant in there, you might potentially not want to score that at a full strength of loss of function as the mechanism of disease and the variant is causing loss of function? What is the mutation spectrum of the disease causing variants? Are there multiple diseases that are posed to be associated with the same gene? Because this framework really, much like a gene disease validity curation, should be looking at a gene, a disease, and a mode of inheritance. And you can have multiple mechanism curations for the same gene, disease, mode of inheritance, or the same gene, a different disease, and a different mode of inheritance. Really, you need to think about that triad. So, what is the mutation spectrum of the disease causing variants? Are most variants predicted loss of function? Is a biallelic predicted loss of function lethal in a human potentially? So you would see like a predicted loss of function in missense. Where are the predicted loss of function variants located in the gene? Are they clustering in a specific region? For example, there's a hearing loss gene where exon 8 skipping is the mechanism of disease. This is not a true loss of function. So you really want to investigate these types of things, or if all the loss of function variants appear to be in the same region, maybe there's something else going on. Um, and then this leads you to what's the mechanism of pathogenicity where you will be scoring. So this is the draft scoring matrix for biallelic loss of function. And for many of our curations, we take previously scored GSEP curations because we know that the GSEPs are experts in those disease areas, and we take the evidence that they have scored and pull from that and score. So for this, because we are scoring mechanism, we do not score at the case level. We actually score at the variant level. However, the variants should be present in bona fide, believable, consistent cases. So really, most of the time when I'm pulling variants to score in a mechanism framework, I make sure that the GSEP has scored those variants and believes them to be in a patient with a biallelic observation, for example, for this biallelic framework that is consistent, has a consistent phenotype with the potential disease for the gene disease mode of inheritance relationship. So first we start by scoring predicted loss of function variants, and these would be meeting PVS1. However, PVS1 says the loss of function is a known mechanism of disease, so we have to ignore the mechanism requirement because that's the question we're asking. But it should be like an NMD plus type variant. So those variants that you would actually go through the PVS1 flow chart to score. And for those, you get zero point 0.2 points. We're kind of in the decimal place land of dosage curation right now. We likely will move to whole points as dosage and SVCV4 harmonize towards whole points, but this is still a draft framework. So this is zero point 0.2 points per unique variant. And if you have a biallelic observation of two loss of function variants in one individual, you can score both of those because it's further evidence that loss of function is a mechanism of disease. You can also add points if there's functional evidence. And we suggest that you really start by scoring as many loss of function variants as possible because you are going to have to perform a full curation on non-loss of function variants to be able to determine A, if loss of function is really the claimed mechanism of disease for those variants, like is there functional evidence that's demonstrating this? And are they meeting pathogenic or likely pathogenic, ignoring the mechanism component? So most of the time for really well-known gene disease relationships, you can max out using the predicted loss of function variants, but you might have to go with some of the very well-characterized non-PLOF variants and rapidly curate them. And those get less points and they have a range depending on their convincingness of like functional evidence suggesting they're causing a loss of function. Then we have an experimental evidence category where we have a knockout model organism with a consistent phenotype and zygosity. And this also has a point range. Oftentimes, I will follow what the GSEP has said in their upgrades or downgrades of a knockout model organism. For this, you really want to be making sure the phenotype and the zygosity are consistent, particularly the zygosity in like monoallelic loss of function curations, which we'll get to in a second. And then if something is really a slam dunk loss of function gene, you might not want to waste your time individually scoring all these loss of function variants. And so you could really get to much of your established loss of function mechanism score if you have a lot of unique loss of function variants. So if you have at least 20 unique loss of function variants and 30% of all of your variants are loss of function, and these variants are found across at least three distinct exons, depending on the size of your gene, you can just start by scoring half a point. And 1 point is what you need for established. So this really takes you pretty far along. Um, if you have a gene that doesn't have a ton of evidence and doesn't have a ton of loss of function variants, but it is autosomal recessive, we really are exploring autosomal recessive disorders having a prior probability of loss of function that's very high, greater than 90%. So we're exploring starting those curations at 0.3 points, thinking that like for a biallelic gene disease relationship, it's just much more likely if you have loss of function variants that loss of function is a mechanism of disease. And then finally, consistency in population databases. So we really want to see absence or significantly reduced presence of homozygous variants of the predicted loss of function variants. Obviously, if they're the ones that are predicted to escape nonsense mediated decay, that's different. And there are tags on that, NOMAD, for example. And other things you have to consider are the age of onset of your condition, of the disease severity. Maybe it's a mild condition and it's later onset, so you would expect higher amounts of homozygous individuals in NOMAD, for example, and also consider the known presence of affected individuals in NOMAD from biobanks. But this will also give you some scores. If you do have some homozygous individuals in NOMAD, for example, this isn't, you wouldn't necessarily be penalized, but you would be getting additional points if you had the absence or reduced presence of homozygous variants. And then for monoallelic loss of function, it's largely the same, but we give different scores for different modes of inheritance essentially for the animal model, the knockout model organism, knowing that for autosomal dominant conditions, oftentimes they have to really push a mouse, for example, and have a biallelic knockout. And so that would actually be reduced points because while that is supportive towards loss of function being a mechanism of disease, it's not consistent with the monoallelic mode of inheritance, whereas you would get higher points for a heterozygous knockout having a consistent phenotype. And then for monoallelic loss of function, we look at LOF scores. So if you have an observed over expected of less than 0.3 for early onset, less than 0.6 for late onset, you would get points there. And also a reduction of loss of function variants with many alleles and nomad, unless they have like a pathogenic or likely pathogenic classification in ClinVar, that which would suggest they might be like a well-known founder variant or something. So that speaks more towards the consistency in population databases for an autosomal dominant condition. And then this is the summary matrix here. So you'll see 90.99 or higher is established. So we have confidence terms for this. So established, likely, and suspected. Um, and uncertain is below that. And you have to have a gene disease relationship of moderate or above to get anything above uncertain, because you just have to have a certain level of evidence to even curate the mechanism here. So to get suspected, likely, or established, it has to be a moderate gene disease relationship. Anything below that's uncertain. Um, for the monoallelic loss of function framework, you can use a ClinGen dosage sensitivity score. So if there's a score of 3, this corresponds to established monoallelic loss of function. Okay, so let's talk through an example here. And this is kind of just a rapid style presentation that I would use to present pilot curations to the GenCC. So this one is RPE65 and RPE65 related recessive retinopathy. So this is a biallelic loss of function curation. I'm sure some of you are familiar with RPE65 and gene therapy for this. So we look at the overview and just make sure that the gene disease relationship is moderate or above, as you would see for this biallelic loss of function curation. So we're just looking at autosomal recessive here. There's also an autosomal dominant assertion. These are strong or definitive, so we're good to go. And then we briefly will talk about what the function of the gene is. This is particularly critical for gain of function and dominant negative curations because much of the experimental evidence is very consistent with the biology. And so RPE65 is a critical protein in the visual cycle, and I'll kind of just leave that at that. Um, you go through the gene level questions. So for this one, it's 14 exons, 533 amino acids. It's a retinoid isomerohydrolase. There's no alternative splicing predicted. The mutation spectrum is loss of function in missense. Asking the question where variants are located, do they cluster, are there hotspots? So there's no clustering observed, but there is a VSEP, ClinGen VSEP for this, and they have a PM1 defined as the active sites, and so those residues are listed here, and then also the ER localization residues listed here. So you might expect maybe higher amount of variation there, pathogenic variation because of those critical sites. And so for this one, even though I likely could have gone and used the loss of function spectrum and just gotten to 0.5 points, I pulled directly from the ClinGen curation and scored directly the loss of function variants within the curation. And you'll see that there are all these different loss of function variants that were scored in the ClinGen curation. And these all get the standard points of 0.2 points. Then looking at experimental evidence, there is a knockout dog model that the GSEP scored, and it has severe vision loss, and the phenotype is very similar to humans, and heterozygous knockout animals are unaffected. So this was also scored in the middle points there. And then for consistency in population databases, there's only one variant that has a homozygous observation in Nomad. All other predicted loss of function variants have no homozygous observations. So we get the 0.5 points there. And this takes us to 1.35 points, which is established by allelic loss of function, as we would expect, as there is a gene therapy for this condition. So let's spend a little bit of time in the gain of function framework now. We come from a similar place, but as you would imagine, we have to have a lot more reliance on the biology and experimental evidence for gain of function and dominant negative frameworks because they are very gene specific, gene disease specific. So here are the questions for gain of function. Many of them are the same. So the size of the gene, number of exons. Um, we're really starting to ask what's the function of the gene. For example, is it a kinase that we would expect gain of function to be a consistent mechanism of disease? We're asking if there's alternative splicing. What's the mutation spectrum in this case, or at least for the gene disease mode of inheritance you're looking at? Are all the variants missense? Maybe there is truncating, not usually for gain of function, it's more for dominant negative. Where are the missense located? Are they clustering, which would be much more common in a gain of function mechanism? Are there any observed hotspots or functional domains? So this is the matrix, the draft matrix for the gain of function framework, where you start by scoring gain of function variants, and um, these should have consistent phenotype. And if you have a GSEP curation, should be scored and believed by the GSEP. However, it's your job to really be looking at the mechanism. So for this, we really have to have strong functional variant level evidence that demonstrates a gain of function, because these largely are just going to be missense variants. So if it's just a missense variant with computational predictors, that's not going to cut it. You have to have functional evidence. And there is a range of scoring depending on the convincingness of the functional evidence. Then we have a pretty extensive experimental evidence section. So a knock-in or a transgenic model organism has a consistent phenotype and zygosity. And this is really showing a gain of function too, that they're not just looking at the phenotype, they're showing that either the variant that they've knocked in or the overexpression is truly causing a gain of function. And there's a range here too. Um, if there's a known function of the gene that supports a gain of function mechanism. So, for example, like I said, if there's a kinase that we know could be overactive in a signaling pathway or receptor, this would support a gain of function mechanism. It has been hard to generalize the descriptions of these types of evidence, so we're hoping to use as many examples as possible in the paper because it is really just very different from gene disease to gene disease. Um, there are studies consistent with the gain of function mechanistic narrative. This is extremely generic, but really just diving into the mechanism and showing why there would be a gain of function. Um, Along the other line, if a knockout animal has no phenotype or an entirely different phenotype, this could be additional evidence that this would maybe be gain of function. And knockout potentially having like just a completely different disease potentially. For a variant spectrum, absence of predicted loss of function variants causing disease, or that the predicted loss of function variants cause a different disease. And really, you should be scoring either the knockout animal model or the loss of function variants causing a different disease, in most of these cases. And then presence of recurrent variants also may suggest that this would be a gain of function mechanism. So let's run through an example real quick so I can give you time for questions at the end. So this is MPL and autosomal dominant thrombocythemia. Some of you may be familiar with this. So we look and see, make sure this is moderate or above. It's definitive by for all the curations. This is showing both inheritance patterns, but this is for autosomal dominant. And so we developed a presentation style that's called a mechanistic narrative, really to speak and help people understand the biology of a gene disease relationship, because much of the experimental evidence is dependent upon this. So MPL is myeloproliferative leukemia virus oncogene. It's a thrombopoietin receptor. It's important for the development of platelets and megakaryocytes and for the self-renewal of hematopoietic stem cells. So loss of function mutations result in thrombocytopenia, which is low platelets, and gain of function mutations result in thrombocythemia, which is high platelets. Just makes sense with the biology. And this is the signaling pathway here. So the size of the gene is 12 exons and 635 amino acids. We talked about the function. For alternative splicing, if you look at GTEx, they're low expression of exons 1 to 8, but it's not predicted that they're spliced out. And we've seen from mutation spectrum that loss of function is thrombocytopenia and gain of function is thrombocythemia. And for missense variants, there observed hotspots or functional domains. There are 4 missense variants, and they kind of are a bit all over the place, 39, 102, 106, and 505. So maybe they don't, we might not have enough information. And this is just GTEx showing you that like the first 8 exons are relatively lowly expressed, but not predicted to be spliced out in the gene model. So when we go to score here, we get 0.4 points. So the difficulty that we run into for gain of function genes is you might see recurrent variants, and we're scoring each one of these variants only once. So you'll see that we have missense variants that are showing gain of function here of different pieces of the biology, but they've been observed multiple times. So the first one was observed 6 times independently. The second one was observed 1 time. The third one was observed 3 times, and the fourth one was observed 1 time. So you can understand how this got to definitive in the gene disease relationship, but we're only scoring the variance once. So the first one and the third one, the GSEP really felt convinced about, and so we scored those at the top of the points. This one was a high population frequency, and they suggested it was a reduced penetrance gain of function variant, so we downgraded this and potentially might not score it at all. And then the GSEP didn't score this last one because it was also present in autosomal recessive cases and it had borderline functional evidence, so we didn't score that one. And then if you look at this, the rest of the points, so for variant spectrum, there's an absence of PLF variants causing disease. They actually cause a different disease, the opposite disease, thrombocytopenia. And then there's a knock-in animal model here that the GSEP scored, and so we scored that. And then for experimental evidence, um, because it's a receptor, it's consistent potentially with a gain of function mechanism, and variants result in increased receptor activity and downstream proliferation. So that's kind of consistent biologically. And so we made it to 1 point here with all of this information, scoring these 3 variants that have been seen multiple times, the knock-in animal, the absence of loss of function variants causing disease, and that it's a receptor. And so we still made it to establish gain of function here. And so, in conclusion, the Gene Curation Coalition, I hope has to show you, I've shown you, is working to harmonize curation terms and frameworks. Our database currently hosts greater than 20,000 gene curations, which is the largest site of this nature in the world, with supporting evidence. And we're currently piloting the semi-quantitative mechanism curation framework that will be adopted in some form by both SVC v4 and then also ClinGen 2. We're in the process of discussing with the steering committee how we would like to adopt this within ClinGen so that we can curate and display mechanisms in a granular fashion on the ClinGen website, which is a very, very popular request. As you can imagine, it is something that would be really helpful to the community, especially as they're doing exome genome analysis. So I want to thank all of the GenCC members and the Broad Geisinger team that works on the website and the database, and then particularly also the mechanism small group that really helped work with me to develop these frameworks that we then took to GenCC to present, and is helping me go through the pilot to really test these frameworks in a robust way. And with that, I will stop sharing and take questions.
Thank you so much, Marina, for a perfectly timed presentation. I saw that there was one question that's just been put in the chat. So, regarding your scoring tables for predicting inheritance patterns, are they going to be published? Are they published in a review article, or did you compile them from various sources?
Yes, so they are draft curation frameworks right now. We might either potentially try to publish them alongside the SVCV4 article or potentially just after that. We do have draft kind of position statements for all of them. So if we don't manage to publish them concurrently with the SVCV4, we'll at least have them publicly available on the GenCC website for people to use, but they're still in piloting right now. So really it's only within the GenCC that we've shared these and the curators within GenCC so they can test them.
Great. And that answer is really helpful to know. So I know that's a question we get a lot of the time when we share draft workflows for V4 of everyone saying, oh, you have this whole component around mechanism of disease. Where can I access that data? So from what I understand from what you just said, that they may not be finished yet and it might not be published as a framework yet, but you do have your position statements available. So
Yeah.
people can be trialing this for themselves.
Yes. Yeah.
Um, any other questions from the group? If not, I do have one more for you. So you kind of alluded to it at the end of your talk about what the plan is for this mechanism curation going forward and how it will be integrated into ClinGen. I guess what is your vision for that? Do you think it's something that individual GSEPs will adopt as an additional curation element that they do as part of their gene curation, or do you think this is a body of work that will be done by a separate group building on what a GSEP's already done?
It's a great question. And I would say, at least in the beginning, the GenCC folks who are also members of ClinGen and the Gene Curation working group co-chairs, Courtney and Aaron, are pushing pretty hard to have a separate group perform the curations and that we have a very large data set right now. What is it, like 3,000 plus genes that exist and have curations but don't have mechanism curations? And it seems like a lot to ask the GSEPs to go back and add those mechanism curations. And you have all of their scoring there. So I was so easily able to like take the scoring from the GSEP and like adopt a mechanism framework. And so dosage is doing the biallelic loss of function. They might move and adopt the monoallelic loss of function exactly and use that too. And then we were thinking of potentially spinning up just a separate group under the variant curation working group of like mechanism curation that would perform like gain of function and dominant negative. That's not to say once we get through this backlog of curations that GSEPs may never do it. And of course, we are hoping that they would pose the mechanism so we would know where to shunt it once that's done. But I think I'm very much in the boat of I don't want to give GSEPs more to do, and they're using their expertise to curate gene disease relationships. But that still has to be seen as we vote on adoption of the framework.
Great to know, and I'm sure lots of people who are involved in GSEPs on that call on this call will be glad to hear that they're not about to have a whole ton more work piled on because I know that gene curation already is a long process. But I think this is going to be so helpful for well for the community generally, but certainly for GSEPs that are already dealing with that issue of the evidence summary statements kind of guide you to perhaps give some suggestion to the mechanism of disease, but if it's not always clear, they don't really know exactly where to go. So this is going to be wonderful. We only have a couple of minutes left, and there doesn't seem to be any other questions in the chat. Just quickly pause and see if anyone wants to add anything. But if not, I think we can wrap up there. So thank you so much, Marina, for that overview of GenCC in the past and what the work that you're doing now is. And just a reminder to everyone, we will be breaking for the next 2 months over the summer and we'll reconvene on September 10th. So see you all then and have a great summer.
Thanks. Have a wonderful summer all. Bye.